Hello everyone, I am Dr. Bhupesh Kumar. First of all, I will thank the organizer of the conference who invites me to this conference to present my work. Right now, I am working as a postdoctoral researcher at the University of St Andrews, Scotland. Here I am going to present my work on torsional properties of spider silk. Title of my presentation is Study of torsional superelasticity and fatigueless response in spider silks. Here is an outline of my talk. I'll start with introduction to the spiders and spider silks, and after that I'll present my work on torsional properties of spider silk, which is not much explored. I'll show the impact of twist deformation on various spider silk, and will show molecular level changes in spider dragline silk when it undergoes extreme twist deformation. Spiders are global citizens. We can find spiders in almost every terrestrial place on Earth. There are almost 40,000 species of spider. Not only that, spiders are extremely diverse. Origin of spiders and its silk are date back to 380 million years. This is order of magnitude higher compared to the existence of human on planet Earth. While working with spiders, it doesn't take much time to realize the importance of silk in spider's life. Spider use silk as a trailing safety dragline, wrapping its eggs for reproduction to capture prey. A garden spider can make 7 types of silks. If you look at this web, here frame and radius are one kind of the silk that is called a dragline silk, and this type of silk is most widely studied by scientists, and the other one, the spiral one, is a capture silk that consists of two parts, filament and sticky glue drop. It plays a very important role in capturing a prey. Here you can see an optical microscope image of captured silk with a glue stick. The garden spider can produce seven type of silk. How does an individual spider can make so many kind of silks? To answer that, we have to look at the spinneret region of the spider. So you can see multiple spiders coming out of the spinneret of the spider. There are many spigot here, and each spigot is connected to its own silk gland which store silk protein. Orb web spider generally can generate the 7 type of silk, and it has 7 silk glands. Each store a different kind of protein. They are tubuliform silk glands which is used to make an outer silk of an egg sac. Aggregate and flagelliform silk gland combine to form sticky spiral portion of the orb web. Pyriform silk gland used to make attachment cement. Cuneiform silk gland is used to wrap the prey, and minor ampullate used for web construction, and major ampullate to form a dragline silk of the spider, and this dragline is also called the lifeline of the spider silk. In this slide, I'll briefly talk about the molecular structure or atomistic model of a spider dragline silk. This silk consists of semi-crystalline fibrinoids where amino acid chains are specially constrained into rigid nanocrystal structures in some region and amorphously free in other regions. Spider dragline silk is a composite material with hierarchical structure. Its primary structure, the amino acid sequence, has been optimized over millions of years of biological evolution. Spider dragline silk is thought to be composed of mainly two proteins, spidroin 1 and spidroin 2. These proteins can be described in a simplest picture as a block copolymer with polyalanine blocks that consist of approximately 8 monomers, and glycerin-rich domains with GGX as the most abundant motif. In spidroin 1, here X denotes a variable amino acid. The loosely conserved motif GGX is repeated approximately 10 times, and glycerin-rich repeats in speed drawing 2 that include GP, GGX and GPGQQ. Here P stand for proline and Q stand for glutamine. The schematic of the hierarchical spider silk structure that range from nano to macro display key structural feature of the silk that includes electron density at angstrom scale, hydrogen bonding strengths, sheets nanocrystal, hetero nanocomposite stiff nanocrystal embedded in a softer semi-amorphous phase, and silk fibrils which assembles into a macroscopic silk fibers. Another proposed model for spider dragline silk structure is a silk core organization with multitude of fibril substructures covered by hard skin. Structures covered by hard skin forms the fiber. The molecular structure here consists of sheet region containing alanine, the red lines here, and glycine, the blue lines, interleaved with predominantly 31 helical parts, which do not contain alanine. All chains tends to be parallel. The crystalline are irregular and strongly dependent on processing. For example, the drawing speed, the rate at which the silk is extracted from the spider. Depending on the method, larger or smaller order domains can be identified as crystalline, shown here with the green boxes, and side and top projection of repetitive model peptide with dihedral angles to show the approximate threefold symmetry in the structure. Now I will talk about linear tensile properties of spider dragline silk. This curve shows here impressive toughness and relative strength of spider dragline silk. The area under this curve indicates spider silk fiber toughness or the energy taken up by this material before it breaks. In terms of strength to weight ratio, spider dragline silk strength is 1 by 1.3 gigapascal, and this is almost equivalent to high tensile engineering steel, which is 1.5 gigapascal. But the spider silk has a relative density that is 1/5 compared to the steel, and it shows its strength is almost equivalent to tensile steel. And in terms of toughness, spider silk toughness is 160 megajoule per meter cube, which is substantially higher than Kevlar, steel and nylon. So here we can say that spider silks are extremely strong and flexible, and based on weight, spider silk is 5 times stronger than steel of the same diameter. If we can produce spider silk at large scale, there are many interesting applications of spider silks in the field of biomedical and technical. Silks can be used to make surgical sutures, bandages, surgical threads. It can be used as an implantable, flexible, optical, electronic devices, and spider silks can be used for making bulletproof jackets, parachute, nets, high-strength ropes, and silk. Recently, researchers have shown that spider silk acting as a optical fiber, it used to transmit the light. Tensile properties of spider silks are well established, but much less is known about the twist properties of the spider silk or torsional properties of spider silk. In this presentation, I'll try to answer the question, how strong is the spider thread under torsion? What is the response of the silk fiber under torsion? And what are the effects of various twist deformation on the structure of the spider dragline silk? We will use Raman spectroscopy to investigate the impact of twist deformation at molecular level. Schematic of our twist deformation experiment is shown here. Silk fibers were mounted vertically with its upper end glued to a rigid clamp fixed on XYZ micropositioner. The lower end of the silk is glued on a rotating clamp attached to a calibrated motor. The experiment was performed at a constant rotation of 158 revolution per minute. The length of the silk filament can be varied from 20 to 100 mm by vertically adjusting the height of micropositioner. We have carefully minimized any residual strain during silk collection and mounting. The wobble in the silk or twisting while twisting silk is reduced by carefully aligning its upper and lower ends. Collimated green laser beam illuminate the twisted silk and resulted laser diffraction due to the silk was observed on a white screen placed at certain distance d from the sample. Using video frame of the recorded diffraction pattern, we calculated critical number of rotations absorbed by the silk before failure, when the silk thread breaks due to twist deformation at certain point, the diffraction pattern disappear. So that tells us the time when the diffraction disappear, and we can calculate the number of turns in the silk thread. In addition, the frame at different time for any number of rotations were extracted and analyzed for transverse compression in the silk due to twist. Performing differential measurement on laser diffraction from twisted silk, we attained a precision of 100 nanometer in the silk diameter change in our experiment. Three different kinds of silk, namely dragline, capture, and egg-case silk extracted from a single spider, Aranus neoscona, were used. We captured this spider from the garden of our institute, and it is an orb web garden spider that produce 7 types of silks. We obtain our samples from spiders kept in laboratory at 25 degrees Celsius and 40% humidity. To collect pristine draglines, spiders were made to jump from a small plastic rod, following which it suspends itself from the dragline. The captured silk were carefully collected from a freshly made orb web. The outer egg case threads are unwoven from the egg sac made by the spiders in plastic boxes. This procedure was standardized to minimize any stretching and twisting during silk extraction. The typical diameter of these silks were around 2 to 6 micron. First, we use standard filaments like 10 micron diameter Kevlar thread and copper wire of 110 micron diameter. These samples are mounted very carefully on the experimental setup, and lower end of these filaments were twisted by the motor about its axis until rupture was achieved. This figure here shows critical number of turn per unit length until the sample break for both metal and Kevlar samples. Shows linear scaling. The critical twists were higher for Kevlar than metal due to its greater torsional elasticity. Here we can see that the Kevlar plot is the green plot and the copper metal plot is here the white dotted plot. To demonstrate the exceptional ability of spider silk to withstand great torsional strains, we also show that in this figure we also show the major critical twist to rupture for dragline, egg case and capture silk of various length 20 to 100 mm. We take threads of different length and measure their measure the twist in those threads until they break, and we plot them as a function of length and the critical turn. This critical turn scales linearly with L. That is, as the length increases, the number of turn absorbed by thread increases linearly until it breaks. And for a dragline silk, the breaking torsional strain comes out to be 350 turn per centimeter. We define here breaking strains as the number of turn a draglines or any thread absorb before it breaks for per unit or per centimeter of length. So these, and we also confirm using some images that these twists are absorbed uniformly by the silk, dragline silk here specifically. Some similar torsional measurements were performed for egg case and capture silk, and corresponding breaking torsional strain was calculated for both. The breaking strain for egg case comes out to be 250 tons per centimeter, and for capture silk it was like 2100 tons per centimeter, which is huge compared to the other samples or other materials, even in the egg case and drag line. So no doubt here that the capture silk shows highly torsional superelastic behavior. To understand the origin of the torsional superelasticity of spider silk, we perform in situ optical measurement to show linear correlation between twist and transverse compression, that is reduction in the diameter of the silk as the number of twists are per unit length are added. A twisted dragline silk produces a complex 2D diffraction pattern, you can see here, with a bright central spot and multiple higher order peaks. To extract the transverse compression, we focus on the central horizontal section in between these dotted lines, horizontal section of the diffraction pattern that corresponds to a single slit diffraction, as shown by this corresponding intensity. Here we take an intensity cut here, and then this shows the intensity cut. So these measurements demonstrate that the width of the first order maxima broadens when more and more twists are added in the silk, and that corresponds to a effective reduction in the diameter or compression in the diameter. And we know that the diameter of the silk is inversely proportional to the width of the first order maxima. So using image analysis to locate first order minima, we obtained a transverse compression with a precision of about 100 nanometer with our setup, and both dragline and capture silk shows linear change in the diameter or linear reduction in the diameter as the number of twists are added per unit length. The maximum compression in our dragline before it breaks was found to be 15%, while in case of capture silk we obtained it up to 25% compression before it breaks. So here we provide that first qualitative measurement of the transverse compression, and we show that the capture silk can compress more and can tolerate more compressibility before it breaks. Further, we observe that the twist response as transverse compression, they are fully reversible. If we untwist the silk thread, the diameter come back to its original form, or diameter starts increasing if we start unwinding the silk threads. The above measurement of torsional rupture limit and transverse compression allow us to estimate maximum potential energy density stored in the silk. Assuming that silk fiber is a homogeneous cylinder with the shear modulus G, the torque exerted by the twisted silk is given by this relation, where J is a torsional constant, G is the shear modulus, theta is the twist angle, and L is the length of the fiber. The breaking energy per unit volume can be calculated by using these relations, and we have calculated it for both dragline and capture silk, which comes out to be 96 megajoule per meter cube for dragline and 1136 megajoule per meter cube for capture silk. As expected, it's more for the capture silk. To summarize this part, we have reported experimental measurement of torsional response of various spider silk by an optical technique, and we find that spider silk exhibit torsionally superelastic behavior where it can withstand great torsional strains before they break, and among spider silk, capture silk was the most torsionally elastic, followed by dragline and egg case silk. Now, in this part, I'll show how spider silk molecular structure change due to twist deformation by using Raman spectroscopy. Raman spectroscopy is a powerful technique to study changes in fiber structure subjected to mechanical deformation. We can examine molecular deformation mechanism of polymeric chain by observing stress-induced band shift due to mechanical deformation. Earlier study shows that intensity bandwidth and peak shift of Raman band is affected by tensile deformation, and we will look for the same under twist deformation. Several bands shift towards lower wave number sides when tensile stress is applied. So this is already reported in the literature. And here I will try to explore the molecular deformation mechanism of polymeric chain in spider silk due to twist deformation. To record Raman spectrum of our twisted and untwisted silk samples, we use a low power helium neon laser to make sure that the samples are not damaged due to laser. And the light is focused on the sample using an 100x microscope objective. Raman spectrum is recorded in the region of 1000 to 2000 wave numbers. When each spectrum is scanned 10 times with an accumulation time of 10 seconds to improve the signal to noise ratio. Here we restrict our study to dragline silk. First we compare Raman spectrum of untwisted dragline with a twisted one. 5 prominent well-assigned Raman bands including amide 1 and 3 region of the silk proteins were observed at 1095, 1245, 1453, 1615 and 1670 cm wave inverse. We observed that amide 1 peak at 1670 is due to carbon double bond O stretch vibration in the beta sheet, and the tyrosine side chain at 1615 cm inverse were mostly unaffected by the twist. This was probably due to the fact that carbonyl group and the side chain are oriented perpendicular to the fiber axis, and also the peak at 1453 which is assigned to carbon hydrogen bond, which is due to the orientation of glycine and alanine residue in beta sheet, is also unaffected by the twist strain. The weak peak around 1550 cm inverse corresponds to the side chains that did not show any systematics in our measurements and could be due to natural variability in the silk. Notably, the peaks at 1245 band due to carbon-nitrogen stretch vibrations in beta sheet and 1095 band due to carbon-carbon vibration in the random conformation, both parallel to the polypeptide change, exhibit significant redshift. This carbon-nitrogen stretch vibration was particularly very sensitive to changes in the internal rotation angle and bond length due to its partial double bond character and its orientation parallel to the fiber axis. Therefore, sensitivity of 1095 and 1245 peak to twist will be used to follow the resulting molecular deformation. We have recorded Raman spectrum of various twisted samples, and on plotting the weak peak position of the Raman bands at 1095 and 1245 cm, we observed a linear shift towards the lower wave number with increasing the number of turns. These Raman shifts indicate that macroscopic shift is directly transferred to the bond in the silk molecule, which changes the bond length and internal rotation angle. The linear dependence of Raman shift with N suggests uniform stress distribution in the fiber and means that the twist-induced bond deformation is higher in the softer amorphous region compared to the crystalline one. What is the fatigue response of a fine dragline silk in cyclic torsion twist? Answer this question is very important to understand the dynamic response of the silk as well as its potential application. We subject dragline silk to a cyclic twist. In one loading cycle, silk was twisted in, let's say, in clockwise direction with n turn per centimeter, and then it was untwisted to 0 turn. This makes one cycle. Two types of fatigue tests were performed. First, a number of twist cycles were fixed to n is equal to 15 and varied the number of twists from 0 to 400, which is close to the breaking limit. The corresponding Raman spectrums were almost identical. Here you can see that the number of each sample with number of twists 100, 300 and 400, and with a cycle of 15 undergoes twist, cyclic twist, and their Raman spectrum is exactly identical to the Raman spectrum of a natural dragline spider silk. Second, we fix the number of twists to 300 per centimeter and increase the twist cycle to 1000. As shown in here, we can see the Raman spectrum, and we can see that all the prominent Raman peaks were preserved and their location was identical to the one for the fresh silk. Particularly the peak, the 1245 shown here, clearly shows reversibility after 1000 cycle. Furthermore, we compare dragline silk surface after 100 cycle, each with 300 ton per centimeter with a pristine silk using SEM imaging. Despite large shear strain in each cycle, SEM image of the worked silk appear to be identical to that of pristine silk. Optical diffraction measurement on the worked silk also confirm the recovery of its original diameter with a precision of 100 nanometer. Clearly, spider silk belongs to a unique class of natural microscale fiber that shows torsional fatigueless response. In concluding part, we find that capture spider silk shows most superelastic behavior compared to other silk. We have demonstrated the linear correlation between twist strain and transverse compression in the diameter and its recovery as well. And we also show that using Raman spectroscopy, molecular structure remains same even after large cyclic twist given to a dragline silk. And in other words, we can say that spiral dragline silk is a biological shape memory superelastic material. Thank you. If you have any question, please feel free to ask on the email address given below.